Okay, so for today, we have some announcements, reminders, and some new stuff going on. So exciting changes. And for the first announcement, next week for Grand Rounds, we will have Dr. Roberto Luis Fernandez, who is a professor of clinical psychiatry here at Columbia. And his talk will be titled Addressing Ethno-Racial Disparities in Mental Health Risk Assessment and Service Access and Delivery. We have 3 reminders that we would ask you to remember every week. The first one is, as a reminder, we are prioritizing questions from trainees such as residents, grad students, clinical or research fellows, and psychologist trainees during the discussion session after today's talk. If you are a trainee, please put the word trainee at the beginning of your question. The second component is that we encourage everyone to post questions at any time during the talk using the Q&A feature at the bottom of the Zoom window. This is separate from the chat, so please put your questions in the Q&A, not the chat. We specifically ask all missions such as education, research, and clinical groups to share their perspective and questions at any time. The third reminder is that we are compiling a list of potential speakers for the upcoming academic year. And by we, I mean me and Jeff Miller and Kate Elkington. We are asking for all of your input, which makes the Grand Rounds a great place for us to discuss ideas and so forth. We aim to have a diverse list of speakers that are covering topics from basic to clinical ideas. And so if you can fill out our survey, Simon will place a link in the chat. And we would love if you can just send us at least one nomination or pass on the link to members of your groups. We would greatly appreciate it. This link is not going to go away. It will always be there. It's permanent. We will monitor it during the year. So if you have speaker ideas that come up over time, please continue to add names for future dates. We value the input of all of the department members and thank you in advance. So today we have something new. We are going to take a survey today. And as we start thinking about transitioning to a hybrid Grand Rounds in person in the auditorium and here on Zoom, we're asking you to vote for the background for the auditorium and here on Zoom. We have 3 options, which I'm going to show you in a second. And Simon will place a survey where you have to vote for option 1, 2, or 3. So I'm just going to share my screen and hope this works. Option one is Lady Liberty that we have. Option 2 is the New York skyline or the Empire State Building that we have it labeled. And option 3 is the GW Bridge with our beloved building in the background and the little red lighthouse. So if you can vote right now, we would love that. And we will do this for the next couple of meetings so that you can put your vote in case you missed a Grand Rounds. As you are voting, the second thing that we are going to try a little bit differently this week is that we want this to be an integrated Grand Rounds where, we're not all stuck on Zoom. And so if you would like to personally ask your question, we are going to try to take attendees out of attendee mode to ask the question themselves. So again, if you have a question, put it in the Q&A. And if you would like to ask the question yourself, please leave a note for me as I try to go through the questions and pull you out of attendee mode so that you can ask Katarina your question yourself. So for today's Grand Rounds, those are the only updates I have for now. I would like to welcome Dr. Katharina Schmack Md Phd. She is a clinical group leader at the Francis Crick Institute and at the University College London. In 2008, she received her MD, and in 2009, she received her PhD from Charité in Berlin. She then completed her postdoctoral training, clinical scientist fellowship, and psychiatric specialization, also at the Charité in Berlin. In 2008, she was awarded a research fellowship from the German National Academy of Sciences. and joined Cold Spring Harbor Laboratory as a research investigator in Dr. Adam Kepik's group. In 2021, from her time in Adam's lab, she published 2 influential papers in Science and in JAMA Psychiatry with Dr. Kepik's. In September of 2021, she also then started her own lab at the Francis Crick Institute. And as a clinical group leader, her appointment is shared with the Department of Psychiatry at UCL. Her research focuses on the biological mechanisms underlying psychosis. Her group investigates the neural circuits and immune processes that give rise to hallucinations and other psychotic symptoms. She uses a cross-species approach, and her lab studies both patients and mice with numeral behavioral tests, computational models, and in vivo measures and manipulations. Their goal is to find a new way to treat brain disorders such as schizophrenia. The title of her talk today is Hallucinating Mice, Dopamine and Immunity Towards Mechanistic Treatment Targets for Psychiatric or Psychotic Disorders. Specifically today, she's going to be talking about how her lab recently established a new behavioral computational approach to capture hallucination-like perception in humans and in mice. And so please again join me in giving Dr. Schmack a warm welcome. Thank you for being here and listening to all that background. And I give it to you to take away. So thank you.
Thank you very much for this very kind introduction. Let me share my screen and my presentation. Yeah, thanks again for this introduction, also for the invitation. I'm really pleased and honored to be speaking to you today. And as Christine already mentioned, I am interested in psychosis. And the reason that I'm interested in psychosis is that, as most of you know, that this condition is characterized by these, I think, very interesting symptoms, which are basically experiences or perceptions of something non-existent. So for instance, in the case of hallucinations, patients typically hear voices where others can't hear any voices. In the case of delusions, many people perceive threats where others can't see any threats. And so to me, this really illustrates the constructive nature of our perception in general, and therefore studying psychosis, in my view, can provide fundamental insights into how we make sense of the world around us. But of course, psychosis is not only intellectually fascinating, it's also highly relevant because it's the hallmark of many different disorders, including schizophrenia. And these are relatively frequent, and we all know that these disorders, what makes them truly devastating is the fact that they start relatively early in life, and more often than not, they do not fully resolve. And unfortunately, this has not dramatically changed over the past decades. So the treatments we have, at least the medications we have, they have somewhat stagnated, and they are all based on mechanisms of action that were discovered by serendipity back in the 50s. And we still lack a mechanistic understanding of how and why the brain and the neural circuitry of the brain goes awry to produce the symptoms of psychosis. And so this frustration drove me from being a psychiatrist really into neuroscience, because in neuroscience, there has been a lot of progress, and we now have really amazing tools at our hands, and I will showcase some of these tools that allow us to study the neural circuitry of the brain in unprecedented detail. The only challenge here is that all these tools are mostly available in mice. So to take advantage of these tools for the study of psychosis, we need to find ways to study psychosis in mice. And that is the overarching goal and theme of my research program, to develop novel frameworks for the study of psychosis in mice and use these frameworks to dissect mechanisms that can guide future treatments. And so today, my talk is structured in 3 parts. In the first part, I will discuss how we started to address one challenge in psychosis research, in biological psychosis research, and that is the challenge: how can we actually measure something that is relevant to psychosis in rodents, in mouse models? And I will introduce a new phenotype that we developed, the phenotype of hallucination-like perception, and show you some data that suggests that this phenotype can serve as a behavioral marker of hallucinations that bridges this translational gap between humans and mice. In the second part of my talk, I will then discuss how we started to use this phenotype to really dig into the neural circuits that are relevant for psychotic experiences. And I will show you some, I think, exciting findings that provide, I think, a mechanistic link between increased dopamine and this phenotype of hallucination-like perception, and therefore confirm the longstanding dopamine hypothesis of psychosis. And at the end, I want to briefly discuss what we are planning to do in our lab here in London, and I will discuss some ideas how we plan to get from these insights to really new treatment targets based on these circuit insights. And I will also briefly discuss how we plan to use insights from the immune system to get to another line of treatments that target the immune system. So one challenge in the study of psychosis in mice is that psychosis is exclusively defined by these very subjective symptoms, such as hallucinations. These are usually only accessible through verbal report. And so previous work in mice or in other animal models has therefore often used some kind of simple behaviors as a measure of psychosis, such as locomotion or the startle reflex after two-tone and after pre-pulse. And these behaviors have provided valuable insights, but I think there's one limitation in these behaviors, and that these behaviors are really far removed from the actual experience of psychosis and of hallucinations. And so today I will introduce the slightly more complex quantitative behavior that we think is more directly captures the experience of hallucinations, and that is the behavior of hallucination-like perception. And I will first introduce a task to measure hallucination-like perception in mice, and I will then show you some data from mice, how we validated this task in mice, and how we related it to hallucinations in humans. But before I start, I just wanted to zoom out a little bit and give you kind of like the full story of my trajectory and how I ended up working in mice. So when I worked as a clinician scientist at the Charité in Berlin, I was already interested in exactly this challenge of how can we actually objectively measure something so subjective as psychosis and hallucinations and delusions. And when I started on this avenue, many theories of psychosis were really focused on these more abstract alterations of higher level cognitive processes. But I think around the time I entered this line of research, there were some researchers around the world that thought that because psychosis is so perceptual in nature, maybe it's might be best described as a perceptual problem. And so we tested this idea empirically. By studying psychosis patients and healthy individuals, and we used some very basic perceptual tasks and combined this with functional neuroimaging. And this is just an example of the kind of results we got again and again. What we observed again and again was that psychotic experiences here on the x-axis were positively correlated with these perceptual phenotypes, both at the level of behavior, but also at the level of neural processing. And so this suggested to me that maybe these very basic perceptual tasks might provide a window into the animal world and might help us to engage some of the processes that are important for psychosis in a mouse brain. And so I was very excited when I got the opportunity to join Adam Kepek's lab in Cold Spring Harbor Laboratory, because he and others there had pioneered the use of these really, of these kind of perceptual tasks in rodents. And so together we set out to model hallucinations in mice. And we started off with a very basic definition of hallucinations. So when we look at what a hallucination is, it is basically a false percept, so a percept in the absence of a causative or correlative stimulus, but that is nevertheless experienced with the same confidence as a quote unquote real percept. And we thought this is something we can operationalize in a task, and that's the task we came up with. So in this task, mice are freely moving in a box, they're running around in a box, and there are three little holes in the walls, three choice ports. And now whenever the mouse pokes into the center port, we play a brief tone signal. And that means that the mouse now has to go to the right to get a water reward, a drop of water. If it goes to the left, it doesn't get a water reward. And this is half of the trials. On the other half of the trials, randomly interspersed, the mouse pokes into the center, and now we do not play a tone. And that means that the mouse now has to go to the other side to get a water reward. If it goes to the signal side, it doesn't get a reward. So that gives us the perceptual choice. So this allows us to measure the first part here, the percepts. What about confidence? And to measure confidence, we did a trick. We simply delayed the reward for an unpredictable interval of time. And sometimes we would even just omit the reward. And so this has the effect that the mouse now never knows when and whether a reward will arrive. And it has to make an active decision how much time it is willing to invest. And the simple idea here is that if the mouse is very confident that it either heard the tone or did not hear the tone, it will wait for longer intervals of time as compared to instances where it is less confident. So in this framework, we can define hallucination-like perception as false alarms. That means trials where we do not play a tone, but the mouse nevertheless reports hearing a tone. And it does so with high confidence, as indicated here by a long time investment. Okay, but just because we call this hallucination, so the first question, of course, is can mice actually do this task? And this was not really clear when we started because most people had worked in rats with this kind of task. So the first challenge I faced is to train mice to do this slightly complex task. And the good news is, yes, if we train mice sufficiently long, they get pretty good at the task. And so what I'm showing you here is a psychometric curve. That means on the x-axis is the signal to noise ratio. So in other words, the volume of the tone that we played over a background noise. And on the y-axis is the percentage that the mice reported hearing the tone. And so I hope that you can appreciate that when the tone was very loud, the mice almost always correctly reported hearing the tone. As the volume decreases, this percentage decreases as well. And now importantly, even if we do not play a tone at all, mice still report hearing tones on a substantial proportion of trials. It's around 15%. So this is good news because this means mice can do the task, and the task elicits false alarms, which is the first part of our definition of hallucination-like perception. So yeah, good news. Now, what about their confidence? Are mice capable of reporting their confidence? And to test this, we first need to have a good idea what a confidence report should look like. And for this, we recur to this idea of statistical decision confidence, which is simply the idea that confidence should reflect the probability of being correct. And because a probability is a mathematical construct, we can use some very straightforward equations to calculate that. And so when we calculate the probability of being correct and plot it in three different ways, this is shown here, we see these predictions of what confidence should look like. Now, when we plot the actually observed time investments on top of that, the time mice invested for waiting for a reward, I hope again that you can appreciate that this looks very similar. And this is true for one mouse, but we see very similar things in all mice that we tested, which is shown here. So basically, what this shows is that time investments do reflect the statistical confidence. So now we have a task that can measure false alarms, and we can, that induces false alarms, and we can measure the associated confidence with that, suggesting that we have everything that we need to measure our hallucination-like perception. Of course, we wanted to know, do mice actually report their perception, or maybe they're doing something completely different? So we ran a few control experiments, control analysis to be sure about this. And so we think that mice indeed are driven by their perception, because we see that what we can observe is not a simple motor bias. So it's not explained by the mouse having a bias towards going to the right or to the left, because we see very similar behavior regardless of whether we train the mice to go to the left for a signal or we train the mice to go to the right for a signal. It's not explained by, so the false alarms are not simple errors. They are something different, because mice don't have errors on the signal tones here, so they get really good. They have specifically these false alarms. It's also importantly not explained by anything that by reward-related processes, because in this task by design, having false alarms is not good for the mice. They get less reward. So there should be no reason for the mice to report a false alarm, because this decreases, as shown here, the overall reward. And we did some further analysis. It's probably not explained by inattention. We don't see any correlation of false alarm rate with any other measures of attention. We also see that false alarms rates are pretty stable throughout the session, suggesting that they are not affected by fatigue or satiation or something like this. And finally, what is just interesting is then when we plot the confidence, the time investments of false alarms and of the correctly perceived signals, the hits, although on average the false alarm confidence is lower, but there's quite a big overlap, suggesting that there are at least some trials where the mice had a comparable confidence or had sufficient confidence that they actually did hear a tone. Okay, so we have this task and we can measure hallucination-like perception, or at least what we call hallucination-like perception. But now, of course, we wanted to know, just because we call it hallucination-like perception, it doesn't mean that it should have to do anything with real experienced hallucinations. So to understand whether this measure was actually relevant for hallucinations, we did a few experiments. The first thing we did, we know from humans, from human literature, that people with hallucinations, they rely more strongly on expectations when they make perceptual decisions. And so we mimic this in mice by having a version of the task where we had different blocks of trials with different amounts of signals. So 30%, 50%, or 70%, meaning that when there were more signals, the mice should expect hearing signals more strongly. And indeed, what we saw that with increasing expectations of hearing a signal, mice also had more false alarms, and they became more confident in these false alarms. And we found, as shown here, that this was driven by a bias in perception. This was not explained by some weird random behavior or random increase in errors. So what this shows is that perceptual expectations induce more hallucination-like perceptions. And given this previous work in humans, we think that this suggests that hallucination-like perceptions might involve some of the processes that are shared with spontaneously experienced hallucinations. So the next thing we did is we did ketamine. So ketamine is a drug that is known that in low doses it can induce some kind of symptoms in people that are somewhat similar to psychosis. It's as good as we can get, I guess, with drug models. And so we treated mice either with vehicle or which was a saline or ketamine. And again, what we found was that under ketamine, mice had more false alarms and they were more confident in these false alarms. And again, and this is really important here, this was not really driven by they did not have just more errors or something. They really had a bias in perception. They had this bias towards perceiving more signals in general. So again, what this shows is that ketamine, a drug that is known to induce some psychosis-like experiences in people, increases hallucination-like perceptions in mice, again, adding some evidence that maybe our task might engage some of the neural mechanisms that are shared with spontaneously experienced hallucinations. But of course, to really know whether something is a good measure of hallucinations or not, ideally, we want to directly relate it to hallucinations. And because we cannot really ask mice whether they hallucinate or not, we translated our task back into humans and came up with this human version that is shown here. It's very similar to the mouse task. People hear tones or don't hear tones and have to say whether they heard it or not. And now we directly ask them how confident they are. And we measured 220 participants from with this task in an online version. And what we found was that when we looked, that the behavior was quite good for, given the fact that this was an online task, so this was a remotely designed experiment, a remotely conducted experiment, and we found that humans showed qualitatively similar psychometrics. They also had some false alarms. They look small here as compared to the mice, but it was a substantial portion of I think 8 or 9%, and also their confidence was quite nicely explained by the statistical confidence model, as you can see here. And now, so we tested these 220 participants, and these participants were from the general population. And now, to get some sense of how to relate this to psychotic experiences, we ran a questionnaire that is designed to measure psychotic experiences in the general population. And many of you might be familiar with this concept, that there is actually quite a high degree of people who occasionally or even more often have psychotic experiences, but do not have the diagnosis of any psychiatric disorder. These questionnaires ask questions like, do you ever hear noises or sounds when there's nothing about to explain them? And then importantly, people are asked to rate how distressing this is and how often they think about these things and how strongly they believe in them. And what previous work has shown is that if you run these questionnaires in the general population, there is a distribution. So the width of the distribution of hallucination-like experiences in the general population is highly overlapping with the one of psychotic inpatients, but obviously there's a bias. So most people don't have a lot of these experiences, but there are quite a few that still have a lot of these experiences. And this is just to show, this is our data to show that we find a very similar distribution as previously reported. So now what we can do, because we have this variation in psychotic experiences, we can relate it to our task behavior in the task. And when we do that, we find that there self-reported hallucinations, they positively correlated with false alarm rate and false alarm confidence. And what was really nice was that this was somewhat specific to hallucinations. So when we looked at other psychopathological dimensions, such as depression or anxiety, we actually found that hallucinations alone explained this task behavior best. And so just to summarize, I showed you a task that can measure hallucination-like perception in mice. I then showed you that hallucination-like perception increases after hallucination or hallucinogenic manipulations in mice. And I finally showed you that hallucination-like perception correlate with self-reported hallucinations in humans. So what this suggests is that maybe hallucination-like perception might be a useful model that can teach us something about the neural circuits relevant to psychosis. And so in the next part, I will show you how we exploited this model to really now generate circuit insights. And we all know that one long-standing idea about psychosis is that there's too much dopamine in the striatum in psychosis. Of course, we know this because most antipsychotic drugs, they all target dopamine receptors, they block dopamine receptors. But also we know this from imaging studies that people with psychotic symptoms, they have an increased dopamine turnover in the striatum. But what we do not know is why too much dopamine in the striatum should lead to these very perceptual experiences in psychosis, such as hallucinations. And we don't know this because we know a lot about dopamine in movement, in reward processing, and cognition, but we don't really know so much about dopamine in perception. And now we have a framework to address this question. And what I will show you, I will first show you a computational model that can kind of give us a theoretical framework for approaching this question. I will then show you some fiber photometry data that provides a correlational link between too much dopamine in the striatum and hallucination-like perception. And finally, I will show you some optogenetics and pharmacological data that suggests that striatal dopamine might be causally involved in hallucination-like perception. So to link any behavior to ongoing neural activity, ideally, we want to have a precise trial-by-trial estimation of this behavior and the governing hidden variables that underlie this behavior. And so to achieve this, we came up with a computational model. We can use this is a very good way to do this. And the computational model is based on this idea that perception is a statistical process that incorporates both prior expectations, but also the current the current sensory evidence. And now in this model, hallucinations emerge when the agent relies too strongly on these prior expectations. So we simulated this idea in a very straightforward model that is shown here. And the model was actually quite good at doing the task. So when we simulated data with this model, we gave them the task structure and we simulated data, we found that the model behaved very similarly to the mice. So here's the model behavior, here's the mouse behavior for the choice and confidence. We also found that the model recapitulated the expectation effect that we had seen in mice. And finally, we saw something really interesting, which it looks complicated, but it's just important to compare the patterns here. So we know from people and from mice that actually that after correct choices, animals and humans tend to repeat these choices, and they do so more if the last trial was very difficult. This is shown here, and the model does exactly this. So this was really nice, a nice validation that the model can recapitulate the behavior in a very close, and detailed way. So now, interesting, the model makes some very precise predictions of how dopamine could potentially be linked to hallucination-like perceptions. So one hypothesis is that if dopamine encodes expectations about reward, then this would lead to more hallucination-like perception, because this would increase errors in general and error confidence in general. Alternatively, dopamine might encode expectations that are more perceptual about the upcoming signal, whether there will be a signal or not. And in that case, too much dopamine would also increase hallucination-like perception by simply increasing signal perceptions and signal confidence. Okay, with these two hypotheses in mind, we now went and measured dopamine in the behaving mice. And to do this, we used a very cool tool, these dopamine sensors. So these are proteins that we can virally express in the brain of mice. And these proteins, they emit fluorescence whenever dopamine binds to them. So what we can then do, we can implant a glass fiber on top of these, of the area we are interested in, to measure this ongoing fluorescence as an ongoing measure of dopamine activity. And based on our predictions, we did this in two regions. One is the ventral striatum, which is known to be important for reward. And the other one is the tail of the striatum, which is a region that receives direct input from auditory cortex and has therefore been suggested to be important for perception. And so what we found, so first we wanted to see what can we measure. So when we know that dopamine is known to respond to reward and stimuli preceding reward, and we observed exactly that in both of these regions. So when we aligned these dopamine traces to the stimulus that was preceding the reward, we found that there were some dopamine phasically responded to that, and the responses were scaled as a measure, as a function of how loud the tone was, which was also a function of how likely a reward was. And now when we looked at the reward responses, we saw that, again, dopamine, there were phasic increases of dopamine after the delivery of a reward, and this was inversely scaled as compared to the stimulus responses, which is roughly in line with this idea of a reward prediction error coding. So this was all nice. This kind of confirmed to us, okay, we are measuring something that we would have expected, so we can measure dopamine. But now our interest was, of course, in these false alarms. And our question was, can we distinguish these? Is there something in the dopamine signal that distinguishes hallucination-like perceptions from other perceptions? And to test this, we now compared false alarms trials with correct reject trials, so trials where the mice also were not presented with a tone, but also correctly reported not hearing the tone. And what we found was in both of these regions, it was quite striking and also somewhat surprising. We found that before anything happened in the trial, so this is the zeros, this is the time point where the stimulus is delivered. So you can see here that before anything had happened in the trial, dopamine was already higher before false alarms. And this is just two mice, but we saw the same in all mice we tested, as shown here. And now when we further subdivided these false alarms into low confident false alarms, which are just, I would say, errors, and high confident false alarms, which are our hallucination-like perceptions, we found that dopamine was particularly high before these genuine hallucination-like perceptions. and this is just another way to plot it, that there was a linear relationship. So the higher dopamine, here on the x-axis is dopamine level at this baseline, on the y-axis is the probability of hearing, of reporting a hallucination-like perception. And so the higher this baseline dopamine was, the more likely the mouse would go on and report hearing a tone with high confidence, even if there was no tone. And again, this looked very similar in both of these regions. Now, when we recur to our modeling, we found an interesting dissociation between the regions. And that is yeah, please don't get lost in the details. Let's just focus on the pattern here. So what we saw was, this is dopamine in the ventral striatum. And when we compare that to the reward prediction from the model, so there's no data in this, this is just a model prediction, we saw that this looked very similar. Whereas in the tail of the striatum, this more perceptual region, dopamine looked very similar to this more perceptual expectation from the model. So what this suggests that in both of these regions, tonic dopamine increases preceded hallucination-like perception, but for different reasons. In the ventral striatum, this might have been explained by an encoding of a dopamine encoding reward. In the tail of the striatum, dopamine seemed to encode more perceptual expectations. Now, of course, we wanted to know, okay, this is nice, but this is just correlational. Is dopamine actually causal for these perceptual alterations we see? And for this, we did an Optogenetics experiment. Optogenetics is another amazing tool from neuroscience. So this is another thing we can express in the brain, that whenever it's a protein or a channel, whenever it's stimulated with light, it increases neuronal excitability. And in our case, we had mice that expressed this in dopaminergic neurons. So whenever we stimulate them with a light, this leads to dopamine release. And so when we now increased dopamine release using this light, we found that this indeed, as we would have expected, did increase false alarm rate and also false alarm confidence. And remarkably, this was rescued by haloperidol, which is shown here. So when we pre-treated mice with saline, we could replicate this effect. But now when we treated them with haloperidol, a dopamine antagonist and antipsychotic, this effect was no longer apparent. So what I just showed you is that I showed you this computational model that gives us some trial-by-trial expectations that can link dopamine and hallucination-like perception from a theoretical point of view. I then showed you that elevated striatal dopamine in both of these brain regions, ventral and tail of the striatum, precedes hallucination-like perception. And finally, I showed you that if we increase dopamine in the tail of the striatum, we can induce these hallucination-like perceptions, and these hallucination-like perceptions are reversible by an antipsychotic drugs. So taken together, I think what this suggests is that, so on a neural circuit level, this this gives us some neural circuit basis for this dopamine hypothesis. And what we think, what our current model is, we think that our results suggest that dopamine might be important for integrating expectations with incoming sensory evidence. So as I told you, the tail of the striatum receives input from auditory cortex, and we think that this might convey these expectations about perceptual expectations about upcoming stimuli. And then it also receives input from the thalamus, which we think might encode these more sensory information. And now what we think is happening is that dopamine is arbitrating between the two, and it's important for modulates the integration of these two inputs. And so when there's too much dopamine, then suddenly perception starts to rely more strongly on these expectations, and that's how hallucinations arise. But what is also, I think, really what I'm very excited about is that I think we now have really a framework that allows us to study the biology of hallucinations, or the biology relevant to hallucinations. And I think that the general idea here is that the assumption that we rely on is that there are neural circuits in the brain, and we can understand psychiatric disorders, but in particular also psychotic disorders or psychotic symptoms, as a specific dysfunction in this in a neural circuit. And now, if we think that these neural circuits instantiate mental computations, we can think that these mental computations, on the one hand, they give rise to subjective experiences, and if they go wrong, then these will lead to subjective experiences that are not that are distressing, such as psychotic symptoms. But on the other hand, these mental computations also give rise to altered behaviors. And we can measure these altered behaviors both in humans and in mice. And in mice, this allows us to then go and look at the neural circuits and inform our understanding of the underlying neural circuitry. So yeah, where do we go from here? And I think it's very nice that we find this link to dopamine, but of course you might wonder what's the point of studying dopamine if we already have drugs that do target dopamine. And I think that is true. But one issue with the current drugs is, and this a kind of a very schematic and naive simplification of the issue, but I think one problem that these drugs have is they do not target the underlying abnormality. So we think that psychosis is caused by an increase of dopamine release, as shown here. But now what these dopamine drugs do, they target dopamine receptors. So they target the downstream signaling of this molecule or this neurotransmitter. And we know there's some evidence that suggests that this over time, because now this cell doesn't get enough dopamine signal, this leads to sensitization, so to the expression of more receptors and more sensitive receptors. And now we actually have two problems. We have too much dopamine in here, but we also have an oversensitive dopamine reception. So now this is something we observe in the clinic very often now when people stop taking their antipsychotic medication. They get what we know as rebound psychosis, because they now get even worse psychosis than they had in the first place. And this also might account for some tolerance effects that we see with antipsychotics. So to overcome this limitation, what we need to do, I think, is to find ways to target the underlying abnormality and to decrease dopamine release in the first step. And that's why we in our lab now are interested in upstream modulators of dopamine release, and we are studying them in relation to this using our paradigm, hallucination-like perception. And one interesting target here are cholinergic interneurons. They interact with dopaminergic. So these are neurons that are first of all, very beautiful, but second of all, they are kind of amazing because they are very sparse. It's just 1% of the striatal neurons, but they have really strong effects on the circuitry. And also they release incredible amounts of acetylcholine, making the striatum the region in the body with the highest concentrations of acetylcholine. So these are truly remarkable. And there's some evidence that they are not functioning well in schizophrenia. And of course, there's some evidence from some clinical trials that increasing cholinergic neurotransmission, or at least muscarinergic neurotransmission, is really effective in treating psychotic symptoms. But the major problem with this strategy is that, of course, because acetylcholine also plays a role in the rest of the body, in the autonomous nerve system, this has really a bad side effect. So it's not really a tolerable strategy. So somehow we need to find the specific locus where this increasing of cholinergic neurotransmission actually does reduce psychotic symptoms. And that's what we're trying to address with our framework by measuring and manipulating acetylcholine and dopamine. Then what we can do, we can then take this one step further and really look for specific markers that are specific to these cholinergic interneurons here, but are not found in other cholinergic regions in the brain and or in the body. And this might give us a handle on how to then target them for another antipsychotic medication. And another challenge we are trying to address is basically, so we now have a model for measuring something that is relevant to psychosis. So we have a phenotype, I think, that is can tell us something about hallucinations and the neural circuitry that are involved there. But what we are still lacking, I think, is a really good etiological model. So we don't have a good way to induce psychosis in mice. And I think that the major challenge here is that it's a polygenic disease with many different etiological factors. So we know that there are a lot of genetic variants that have been identified in the US that all there are also a lot of environmental factors that have been identified to increase the risk for psychotic disorders. But the issue here is that each single of these factors only increases the risk for psychosis by a small amount. So if we were to model one of these factors in psychosis, in mice, then we would need a huge cohort of mice to actually get only one mouse with this phenotype. And even more importantly, even if we model a genetic variation that has higher penetrance, we still have the challenge that we need to know when we should look at the mice, because psychosis is a state. It's not really, so we see it at some point in life. And so to overcome this, I think what we need to do, we need to find a way where all these different factors, or some of them at least, converge a common causal endpoint. And obviously the brain is the ultimate endpoint of this. But I think there is good evidence that the immune system might be one endpoint, one biological endpoint that is one step closer to these factors, at least in some cases. And so to just give you a little bit of evidence for this claim, I think there's increasing evidence that the immune system might be overactive in psychotic disorders. So we know that from these GWASs that much of the genetic risk has been mapped to regions that are in immune-related genes. We also know that there's an epidemiological association with autoimmune disorders. So psychotic disorders are associated with increase the risk for autoimmune disorders and the other way around. We also know that prenatal infections, but also postnatal infections increase the risk for later psychosis. And this has been demonstrated in the COVID pandemic, where it very rarely, but it has been observed that COVID can induce some very acute psychotic episodes, which has been known before from some other viral infections, but just proof of principle that somehow the immune system can go, if it goes wrong, it can induce psychosis. We also know that people with psychosis have an increase in pro-inflammatory cytokines. There is some evidence that antipsychotics actually also act on the immune system, suggesting that maybe some of their effect might be attributed to their immune modulatory effects. And then there are these are just case reports, but there are case reports that suggest that stem cell transplants can transmit schizophrenia. So there's one case report from a patient who had chronic schizophrenia and then needed a stem cell transplant, basically a new immune system from another donor. And when he got this immune system from a person without schizophrenia, this patient's schizophrenia also completely remitted. And this case is even more impressive together with another case where a person who did not have any psychiatric diagnosis, this person needed a stem cell transplant and received a stem cell transplant from someone with schizophrenia. And after that, this person deteriorated and also got symptoms that were very similar to schizophrenia. So that's one avenue we're trying to pursue. And so what we are doing, we are collecting tissue from patients, both serum and cerebrospinal fluid, and then do some unbiased profiling. Of course, one challenge that we always have in this kind of studies, we all know that patients are very different from healthy individuals, not only because of their psychotic symptoms, but because of many other factors such as smoking, all these factors that are associated. And so it's always difficult to actually establish causality. So even if we find something here, how do we know it's causal? And I think that's where we can now come in with our mouse model and then try to mimic what we find here in our mice and see whether this induces these behavioral abnormalities that are similar to hallucinations. And so just to end on my kind of big vision, so I hope that what we will achieve in the next decades, and I'm actually quite optimistic that we together will achieve that. I hope that we will get to a point where we will have mechanistic treatments for psychotic disorders. On the one hand, I hope that we will have circuit-based treatments, that we can identify circuit abnormalities that we can target with new treatments. And on the other hand, I hope that we will get, by looking more at the immune system and understanding also more about the immune system and in the brain, we will be able to identify immune mechanisms that are specifically affected in psychosis, and this will allow us to then develop targeted treatments for that. And with that, I would like to thank, so this work, most of the work I did was conducted in the lab of Adam Kapich in Cold Spring Harbor Laboratory. So I would like to thank him and his entire lab. Very collaborative lab, so most everyone here in the lab had a substantial contribution to this project. And this is my small new lab and the Francis Crick Institute. And with that, I look forward to your questions.
Thank you so much for that talk, Katarina. It was wonderful and broad,
Thank
and
you.
I greatly appreciate it. So today is the first day we're going to try this, so bear with me. We have a question from Christoph Kalendonk, and so I'm going to start off with Christoph since he invited you. Try this and see if it works. Oh, he's there. Okay. Christoph, whenever you want to ask a question, you can go ahead. We can't hear you yet, just FYI. Okay, maybe when Christoph is trying to get his video or audio to work, I'll read you some other questions if that's okay, and then we can go.
Yes.
Further. So we have a question from an anonymous attendee, and the question is, can you clarify what is meant by time investment and how/why it is used as a proxy for confidence? Why does more time on the task equate confidence, or is it the other way around?
Yeah, thanks. Thanks for this question. So time investment simply means the time, the seconds, the mice wait. So when the mouse makes a decision, it then goes to the left or to the right to get their reward. And time investment simply means the time the mouse then waits until it will abort the trial and start a new trial with a chance for a new reward. And so intuitively that makes sense, because intuitively, if the mouse is very confident that it was correct, it will wait very long, because it's like, oh yeah, I got this right. I know a reward will come, so it will wait longer. But there's even a stronger quantitative explanation for this. So we know we can use these mathematical model of statistical confidence, and we see that the time investments we see in mice, we also have some other data from humans, they are very, very statistically optimal in the sense that they really reflect the probability of being correct. So it means more time means more confident because they know that they will they kind of expect to get this reward. I hope I answered this question. There's a lot of there are several papers from Adam Kapic's lab around this construct, and it's really, I think it's really nice, but it's hard to explain in just 1 minute.
Thank you so much. Christoph is here, so I hope his audio works. He's going to ask the next question.
Yes, so I got kicked out somehow, and then I rejoined. This was a great talk, fantastic talk, and I think it's so exciting that we now have a handle to study positive symptoms in mice. I think it's very, very exciting. I had a question regarding dopamine. So you showed when you increase signal expectation, you get more false alarms, and you show that when you enhance dopamine, you get more false alarms. Is there the possibility that the first may cause the second bias, so that the increase in signal expectation may lead to more false alarms via enhancing dopamine?
So yeah, whether increasing so that kind of the optogenetic manipulation increases expectations and thereby leads to more dopamine, is that?
No, I thought that if you change the rate of tone versus non-tone trials, whether this somehow is signaled via dopamine, and there may lead to more false alarms.
Yes, we haven't tested that for a very technical reason, because it's really difficult to measure this with a fluorescence-based signal that decays over time. So it's really hard. So, but we are thinking about this. We are coming up with better models to test this. So it's similar with ketamine. We also want to understand whether ketamine acts on dopamine, but because our signal is in this baseline dopamine, we are a little limited with fiber photometry and what we can actually, what kind of manipulations we do. So we're really limited to kind of trial by trial manipulations, but we're working on that. Thanks.
Thank you, Christoph. I'm going to move you back to attendee if you don't have any more questions, but you can ask.
Oh, I have another question.
Go, you're on then.
But I don't want to monopolize here. No, of course, then one question is, I mean, in the end, you think the imagination would be that you get this a nice neuronal response in the auditory cortex at a time when there was actually no real signal. There was no tone, but you still get kind of this tone. So that means somehow the stratum has to go back. Do you have any idea how the dopamine in the stratum would basically then kind of in the end affect auditory cortex function?
Yeah, either it has to go back or it has to kind of reinforce something that is in there. But I mean, we all think about this in loops, right? I mean, it's all this loop that goes back and forth. But we still, I mean, that's one of the plans. We want to look at whether we can see these expectations in auditory cortex, because this will help us to then understand also the temporal relationship, whether it's just the expectation that is kind of amplified by dopamine, or whether dopamine somehow impacts this expectation. I'm also not putting all my bets on auditory cortex. We might also find it in other regions like frontal cortex or even hippocampus. So I'm not 100% sure that it has to be an auditory cortex, because I mean, imaging studies show this big cluster with spontaneous hallucinations that encompasses auditory cortex, but it's kind of spread out and goes into insula and prefrontal cortex. So we'll see what we find.
Thank you, Christoph, very much. So I'm going to move you back now. So we have a question from Arturo Torres, who I'm going to bring in also right now, and hopefully it will work this time. Arturo, if you can hear us, you are good to ask whenever you want.
Hello.
Hi.
Hi. So great talk. It was very, very interesting. And my question is somehow also related with some of the aspects Christoph was mentioning. But in my case, it's more focused on really the training and the fact that you were testing multiple probabilities of having the tone or the no tone, and then you were relating this with prior expectations somehow. And I was wondering if, for instance, you have some trials that it's a tone trial, but with low signal to noise ratio, the animal may be perceiving this trial as a no tone trial. So I was wondering, how did you, or if you could control for the exact probability of tone, no tone trials?
Yeah, no, I see what you mean. So what we controlled for, yeah, you're right. We just controlled like the stimulation, and we did not control the perception fully, because that's what was our readout basically. So you're right, the probabilities shift, like there will be more, the perception will be changed. So we cannot match the perception because that's what we're trying to change. But you're right that this does not fully match, like with, for instance, how often the mouse goes to the left and how often it goes to the right, and also how often it gets rewarded on the left or on the right, because that changes as soon as the perception changes.
Right.
And we see it does change, like even for the trials where we had signals, but they were kind of low signal to noise ratio, we see this shift similar to the false alarms. So we see the shift on all the trials, but it scales as a function of, so the clearer the tone was, the less important was this prior, what we call expectation. So the expectation had a particularly strong effect on no tone trials and on these difficult low signal to noise ratio trials.
Right. And also related with that, so I was wondering, do you think that the amount of false alarms that you get in a session may be also related to the fact that just having more uncertainty in the tone, also it links with having more fails in the no tone side than in the tone side? So having these trials with low signal to noise ratio, the animal may go to the no tone side, and then it will actually go through almost a reward expectation value.
Yes. So certainly, like, uncertainty is very important for these false alarms. Like we only see, like it's true. Like if we have a clearer like if we reduce uncertainty in this task, then we also reduce false alarms. So that's a factor. Yeah, I think that's probably also, that's why we also think that, I think that these false alarms are reflections of kind of some kind of expectations that just build up from the previous trials.
Right.
And that's what we see in our computational modeling. So I think there needs to be uncertainty so that a prior can ever have an impact, because otherwise, if the sensory information is completely clear, then you will just go with the sensory information.
Right. Okay. Yeah. Thank you.
Thanks.
Okay, I am going to bring in someone else to ask a question, and as they're coming in, I will read you some other questions if that's okay. So Meg Haney has no question. She just wants you to know that she found this talk amazing. So that's from Meg. And then we have a question from an anonymous attendee who asked, or who said first, such interesting work. Do you have more temporal information on the increase in dopamine? How long before the false alarm can you detect the elevated dopamine? Or if you increase dopamine far in advance of the trial, does it still lead to increases in false alarms?
Yeah, that's a great question. So because of our design, we don't have a lot of data on that because we cannot really go back in time, but there are some trials where we can go back in time. It's just that the error becomes really big, but we see that these increases go back really far in time. Until we looked, I think we looked up to 10 seconds. So it seems to be really long, like a bias that is there, yeah, encoded as a state. We have not tried the stimulation. So our stimulation protocol was aimed at mimicking this tonic thing. So we basically stimulated continuously, like with a frequency, with a tonic low intensity protocol. So we cannot really answer this question in this data set, but I would expect that there is a kind of lasting effect that you will probably mess up behavior, even if you, over the next seconds, or minute. I think these are slow effects.
Thank you very much. We have now Guillermo Horta here. So I think he's well equipped to ask his own question. And Guillermo, take it away, whatever you want.
Sure. Thanks so much, Katarina. It was super interesting talk, and I really think this is super innovative, paradigm shifting work. I have one more specific question and one general question. The more specific question is, where do you see your work positioning candidate models related to reward, the role of alterations in reward dopaminergic signaling in psychosis? Whether you're confident enough that you can basically rule out those models, or what is there to be done to to evaluate their potential role? And then the second question is more general, and it's about, what do you see the role of human cognitive neuroscience in psychosis, and how do you see that kind of synergizing with the type of work that you do?
Yeah, no, maybe I can answer the second question first, because I'm thinking a lot about this, and I think I think the beauty of these, we call it computational psychiatry, but it's kind of, you can call it whatever. I think what it is that we have a behavior that is quantitative and that is driven by an algorithm that we can quantify with a model, like that we can calculate. I think that's the beauty of this. And the beauty of this is that we can do this in humans and in mice with the same behaviors, and that kind of increases our confidence that we are actually looking at something similar. If we see the same algorithms at play in mice and in humans, then we can translate back and forth. And I think in our specific case, I'm very excited about the idea. So we have some preliminary data where we actually mess with acetylcholine, and we also see an influence on hallucination-like perception, but it looks slightly different. So we see false alarms go up, but we see like confidence stays the same. So there is like the subtle difference in the behavioral phenotype, and the hope would be that we can, that's my hope, that we can take these phenotypes and apply them to people and kind of map them onto these underlying alterations that we find in mice. And then this might be a way to kind of mechanistically informed stratify patients for treatment. So that's kind of the bigger picture, and I think it's very important. And the same might be true for neuroimaging. If we see in our mice that it's one region that is important or gives rise to one kind of phenotype and not the other, then this is something that we can directly try to then translate into neuroimaging and use this for treatment development. So I think that's actually I'm very excited about this possibility, and I think that the years to come will show whether it holds up and whether we can actually do this. And your first question was about, can you just give me one prompt?
About the role of alterations in reward processing in psychosis.
Yeah.
I mean, I'm thinking about the salience misattribution models that I see as more reward related. They're different versions, but just your thoughts on that.
So, I mean, definitely, like, our results cannot be only explained by salience attribution, because there is no explanation for why there should be this asymmetry between like one side and the other side, because they are potentially equally rewarded. On a larger, one way to answer this question, like from a little bit bigger picture perspective, I think that, of course, we have all these cognitive domains in our heads, and we think reward and perception, but I think very much that perception and reward are actually not separable. So I think that our perception is made to perceive only stimuli that will be relevant, that in some way, that have the potential to be rewarding, and that will guide our actions. And I mean, sometimes these actions might be aborted before. If we kind of imagine something, then of course we don't have an observable action. But I think that's the purpose of perception. And so I think on a more philosophical level, we cannot fully disentangle like perception and reward. It's not this kind of perception, then valuation, then reward. But I think this is also something that I hope that the next years will, on a conceptual level, help us to make some progress there. What's an alternative model for brain functioning that has not these kind of step sensory processing and going on, but kind of accounts for the fact that they are linked and that they influence each other, that actions and reward act back on perception. That is a kind of a loop. I think there's some very exciting active inference models that kind of try to catch up or formalize this idea of that it's not a linear progress through a hierarchy, but it's kind of a feedback loop.
Thank you.
Thank you, Guillermo, also for joining us. So I'm going to read you another question as I bring someone else in. Okay, so we have a question from Michael Grunenbaum. He thanks you for a great talk, and he would like to know, how would you reconcile the data you present with clozapine, which is arguably less dopamine blocking, being one of the most effective antipsychotic drugs? So can you comment?
Yes. So first of all, I don't know whether what we see here captures all psychosis cases. And I think it's very conceivable that there might multiple routes to psychosis, that some of them might not be important for clozapine. But even if it's all through dopamine, then of course there will be upstream mechanisms and downstream mechanisms that are not dopamine, and that might be similarly, like that might be perfectly affected by clozapine. I think, yeah, we just don't have like the complete picture here, and we don't know how pervasive this mechanism is actually in patients. Yeah, if I can digress, there's actually some very interesting evidence that clozapine also has a strong effect on the immune system, which is something we've become very interested in. So this might be another explanation, but yeah, that's very speculative at this moment.
Thank you very much. We now have Alex Harris joining us. So Alex, take it away.
Hi, Katarina. So,
Hi.
So I guess this sort of follows nicely on Guillermo's question. You show very nicely that the dopamine, excess dopamine in the tail of the striatum correlates and perhaps causes these hallucination-like perceptions. So do you think that in psychosis, in clinical psychosis, that it's limited to that circuit, or is there a general problem of excess dopamine that could potentially be in other circuits and cause other domains of psychiatric symptoms like dysfunction in reward seeking, or cognitive inflexibility, or other symptoms associated with psychosis?
Yeah, I definitely think that this is a possible, like I don't have a clear idea about, like, I don't have any data or any evidence to argue for one or the other. Basically, so I have to say, when I started this project, I didn't think that we would find regional specificity in this striatum. I kind of viewed the striatum as this one big neuromodulatory structure. So I surprised myself by finding that we have these differences in these two regions of the striatum, which are quite far apart. So it's fair that they are different, but still it's not necessarily expected from a neuromodulator that it encodes something so specific. So I think it's very, feasible that dopamine might be hyperactive in general. And I mean, I think the general idea is, like, I mean, as you know, and many others, there's this idea of a cortical hypodopaminergia and a striatal hyperdopaminergia, but I think this has not been really settled. Like, I think it's still possible that there might be also too much dopamine in the cortex, and that explains cognitive symptoms, and in other region of the striatum, these reward symptoms. So yeah, I completely agree. And we know schizophrenia or psychotic disorders are complex, like they have all these complex different symptoms, different symptom clusters in different people. So this might explain also the variability that where exactly that is.
Thank you.
Thank you, Alex, very much. So I'm going to put you. Okay, so I'm going to bring someone else in and ask you another question in the interim. So we have a question from Rachel Marsh. She agrees with Meg about being a great talk. She said, what a great talk, and your excitement about this work is clear. I'm just wondering, have you or will you look at this in relation to the development of psychosis risk?
We don't have any concrete plans, but obviously that would be very important. I think now we try to start on the extreme, like to kind of get a handle on that. But of course, we would be very interested in seeing how this progresses over time, and how these things might be pre-existent. I mean, our data in humans shows that it's basically from a normal population, shows a relationship to psychotic symptoms in a normal population, which is an indicator of increased psychosis risk. Not one of the strongest one, but it is one. So I think there is kind of a reason to believe that what we find is probably an extreme endpoint, but it's probably already there during development. So many things to do.
Thank you for answering that. We have now Alex Janowski joining us. I'm going to ask to unmute. Hopefully this will work. And he has a question for you as well.
Hi, Katrina. This really is a paradigm shifting work in terms of trying to model hallucinations in mice. But actually, I was hoping that you could dig in a little deeper into something related to what you just mentioned in answering Alex Harris's question. And that's, you sort of use hallucinations and psychosis a little bit interchangeably, talking about the striatum. But the data on dopamine hypofunction and the PFC, it's not that little, right? Referencing like Kurosawa's work in mice and humans, postmortem work from Pittsburgh, imaging work from Anissa Abidorgam. So I'm hoping if you can just try to help us, especially as you talk about potentially down-regulating dopamine release as a possible strategy. Maybe you can place your work a little more mechanistically in the context of dopamine hypofunction in the PFC.
Yeah, so I think the reason why I haven't even started thinking very thoroughly about the PFC is that it's really hard for us to measure dopamine in PFC. At least it's not as easy as it is in the striatum, because the concentrations are much lower. Yeah, I mean, I think that it's a matter of looking at this, and we now can do this. So we could look at projections that specifically go to the PFC, and we can do this work, and we can see whether this is important for our psychosis phenotype. But of course, we can also then look at cognition and other things. So I don't have any strong hypothesis. I think we just don't know yet what to expect, and whether like it's necessarily dopamine is too much dopamine in the striatum is necessarily associated with not enough dopamine in the prefrontal cortex, whether these are two different things that can often co-occur. So I think we just don't know this.
Thank you, Alex. I appreciate it. Okay, we have two more questions. So the first one is from Jeff Miller, and he had the same question I had. So it's going to be a Jeff Christine combined question. He said, that was an amazing talk. Can the mouse model or the behavioral assay that you developed help in a way to screen a broad library of compounds for antipsychotic effects? And my part of the question is, you talk about ketamine, and obviously there's this huge field of like, what are the molecules or the components of ketamine that are driving some of the hallucinogenic effects. And so can you start teasing this apart or screening with your test?
I wish I could say, yes, we can, but I think I need to be honest, and our task is not really a high throughput task. So we train these mice super long, and they are trained like 3 months. So and we are limited in terms of just manpower and equipment. So I think this task is particularly useful for looking at neural circuits, because it allows to kind of monitor the processes that go on, but it is not such a good task for screening. But something that would be great for us is to see, maybe we can simplify this task, or we can also find out like how it relates to these more simple phenotypes, and kind of become more confident of what combination of these simple phenotypes that we could then use for this might be a good reflection of these processes. So that's the hope. But yeah, I think as of now, it's not really suited for it. It wasn't made, and it's not really suited for these more high throughput screenings. And with respect to ketamine, yeah, I think it's a very interesting question. And also with respect to psychedelics in general, and I have one colleague who's also co-author on the paper who just started his lab in Germany, and he will be looking at some of these questions related to this with psychedelics, using a visual version of the task. But exactly, like, is this task also a good measure for these more psychedelic perceptual experiences, and can we use this to kind of tease them apart from the other effects of these compounds?
Thank you so much. Okay, we have a question from an anonymous attendee who said that given that many folks with primary psychotic disorders also experience extremely debilitating negative symptoms, and that the negative symptoms are associated with downregulated dopaminergic activity in the mesocortical tract. So for individuals with positive and negative symptoms, could there be a way to specifically target striatal and mesolimbic pathways?
Yeah.
Can you talk about that?
Yeah, great question. And I think there are ways. I think there could be ways, and I think striatal interneurons are probably one good target for that, because they are only in the striatum, so they affect dopamine release, the axonal dopamine release there. So that might be one way, or of course other neurons that kind of are specific to the striatum and affect dopamine there. And then, of course, another possibility is that we could now, with all the tools available and with people doing a lot of molecular characterization of circuits, of course, there is also a possibility that these projections, they differ in their molecular structure, but also in their input. So I'm sure that just neuroscience will give us some answers to these questions, and will kind of guide us like towards more specific targets in these circuits.
Thank you so much. We have one more final comment, and I agree. Alex Harris is messaging me saying that he wouldn't discount your task as readily for testing, compounds as high throughput, because you could, in theory, right, test the same mouse, on different metabolites, ketamine or something once they're trained up. So I agree with that. Yeah, that's true. You could sell it, I think.
I have been contacted by some people about that, but I always told them, no, don't bother with me. It's too complicated, but I will reconsider now.
Now you can call us. We're on your side. So the last comment for today, I would like to invite Dr. Blair Simpson, our interim chair. She wants to make a final comment, and thank you again for putting up with our survey and all of this trial and run today with people coming in, and you gave a wonderful talk. So thank you, Blair.
Thank you so much, Katarina, for talking today. And you know, we didn't really have a chance to talk so much about our department, but as I said, it's 150 principal investigators, and you got a lot. We have 30 people who study animal systems of one sort or another, and you got a lot of questions from them, but your talk also tapped into people who do clinical trials and that type of work. And I only want to say that I also found it incredibly exciting, not only because of a mouse model of hallucinations. I work on OCD, and one of the issues, could you ever come up with a mouse model of obsessions, right? So you're sort of showing an approach to a symptom that we see in patients. And the question is could you ever model that in a mouse? And you're suggesting we could. But to Guillermo's point, the other one was the use of computational modeling to actually translate between the animal system and the human behaviors. I find that actually really exciting, because then now you have something that can drive human imaging studies as well as human treatment studies. So I actually think your work fits beautifully in the department's sort of portfolio from basic science to translation. And we want you to make this mouse model high throughput, because for any of the clinicians listening, how desperately we need new treatments for our patients. And the problem is how are we going to find them? And obviously one way is through better experimental systems like yours that potentially give us new routes into the phenotype and the mechanisms that push to new treatments. So with that, thank you so much. She's joining us from London, so have a good evening.
Thank you. Thanks for the
you.
invitation.
Bye-bye.
And it was fun. Thanks for all the questions.